Cephalopod ink is a dark pigment released into water by most species of cephalopod, usually as an escape mechanism. All cephalopods, with the exception of the Nautilidae and the Cirrina, are able to release ink. The ink is released from the ink sacs and is dispersed more widely by accompanying its release with a jet of water from the siphon. Its dark color is caused by its main constituent, melanin. Each species of cephalopod produces slightly differently colored inks. Generally, octopuses produce black ink, squid ink is blue-black and cuttlefish ink is brown. A number of other aquatic mollusks have similar responses to attack, including the gastropod clade known as sea hares. Inking behaviors. Two distinct behaviors have been observed in inking cephalopods. The first is the release of large amounts of ink into the water by the cephalopod, in order to create a dark, diffuse cloud which can obscure the predator's view, allowing the cephalopod to make a rapid retreat by jetting away. The second response to a predator is to release pseudomorphs, smaller clouds of ink with a greater mucus content, which allows them to hold their shape for longer. These are expelled slightly away from the cephalopod in question, which will often release several pseudomorphs and change color in conjunction with these releases. The pseudomorphs are roughly the same volume and look similar to the cephalopod that released them, and many predators have been observed attacking them mistakenly, allowing the cephalopod to escape. Furthermore, green turtle hatchlings that have been observed mistakenly attacking pseudomorphs released by octopus botki have subsequently ignored conspecific octopuses. However, many cephalopod predators have advanced chemosensory systems, and some anecdotal evidence suggests that compounds such as tyrosinase found in cephalopod ink can irritate, numb or even deactivate such apparatus. Unfortunately, few controlled experiments have been conducted to substantiate this. Cephalopod ink is nonetheless generally thought to be more sophisticated than a simple smoke screen. The ink of a number of squid and cuttlefish has been shown to function as a conspecific chemical alarm. Octopuses have also been observed squirting ink at snails or crabs approaching their eggs. Chemical composition. Cephalopod ink contains a number of chemicals in a variety of different concentrations, depending on the species. However, its main constituents are melanin and mucus. It can also contain, among other things, tyrosinase, dopamine and L-DOPA, and small amounts of free amino acids, including taurine, aspartic acid, glutamic acid, alanine and lysine. Protective mechanisms. Cephalopod inking in the sea hare Aplysia californica, provides protection from spiny lobsters, a major predator of the sea hare, by means of three mechanisms: chemical deterrence, sensory disruption, and phagomimicry. The typical defense response of the sea hare to a predator is to release of chemicals such as free amino acids, ink from the ink gland and opaline from the opaline gland. Chemical deterrence involves the release of toxic chemicals which are noxious to predators and rapidly dissuades them from feeding. Ink creates a dark, diffuse cloud in the water which disrupts the sensory perception of the predator by acting as a smokescreen and as a decoy. The opaline, which affects the senses dealing with feeding, causes the predator to instinctively attack the cloud of chemicals as if it were indeed food. Use by humans. Cephalopod ink has, as its name suggests, been used in the past as ink. Indeed, the Greek name for cuttlefish, and the taxonomic name of a cuttlefish genus, sepia, is associated with the brown color of cuttlefish ink. Modern use of cephalopod ink is generally limited to cooking, where it is used as a food coloring and flavoring, for example in pasta and sauces. For this purpose it is generally obtainable from fishmongers or gourmet food suppliers. The ink is extracted from the ink sacs during preparation of the dead cephalopod, usually squid, and therefore contains no mucus. Recent studies have shown that cephalopod ink is toxic to some cells, including tumor cells. See also, cephalopod. References. External links, an article on harvesting squid ink.